Welcome to another session. Today my topic of discussion is numerical taxonomy. In biological systematics, numerical taxonomy is a classification system which deals with the grouping by numerical methods of taxonomic units based on their characters. Numerical taxonomy may be defined as the methods of grouping of taxonomical units by numerical methods. It is an approach to biological classification which uses overall similarity to assess relationships and is also called as phonetics and taxometrics. The concept of numerical taxonomy was proposed by Michael Adamson, a French botanist, planned to assign the numerical values to the similarities between organisms, and he proposed that the equal weightage should be given to all the characters while classifying plants. He used as many characters as possible for the classifications, and these classifications came to be known as the Adamsonian classifications. Later on, Robert Sokal and Peter Smith in 1963 published a book named as Principles of Numerical Taxonomy and attracted the scientific communities toward the numerical taxonomy. After that, he would in 1967 defined the numerical taxonomy as numerical evaluation of the similarities between groups of organisms and ordering of these groups into higher ranking taxa on the basis of these similarities. Later in 1973, Smith and Sokal divided the fields into phonetics and cladistics. In phonetics, the classifications are based on the patterns of overall similarities, and while as in cladistics, the classifications are based on the branching patterns of the estimated evolutionary history of the taxa. Thus, nowadays, the numerical taxonomy is more appropriately referred to as phonetics, which refers to the applications of various mathematical procedures to numerically encoded character state data for organisms under study. Thus, the applications of Adamsonian classification principles and the use of modern methods given by Robert Sokal and Peter Smith and electronic data processing techniques have helped in the evolution of several new classifications of plants during past few decades. Thus, the numerical taxonomy is the analysis of various types of taxonomic data by mathematical or computerized methods and numerical evaluation of the similarities or affinities between taxonomic units, which are then arranged into the taxa on the basis of their affinities. Now the aims and objectives of the numerical taxonomies. The main aim or objectives of the numerical taxonomies is, the first one is to determine phonetic relationship between organisms or taxa. The second one is to classify organisms using numeric algorithms like cluster analysis rather than using subjective evaluation of their properties. And the third one is to illustrate the degree of relationship or similarities between two or more organisms. Now, the aspects of the numerical taxonomy. Numerical taxonomy involves two aspects. The first one is construction of taxonomic groups. In numerical taxonomy, first, individuals are selected and their characters spotted out. There is no limitations to the number of characters to be considered. However, the larger the number of characters, better is the approach for the generalization of the taxa. The resemblance among the individuals are then established on the basis of character analysis, which can often be worked out with the help of computers, the accuracy of which depends on the appropriateness in character. The best way to delineate taxa is to utilize maximum number of characters with similar weightage given to all of them. The second aspect of the numerical taxonomy is discrimination of the taxonomic groups. When the taxonomic groups chosen for the study show overlapping of characters, discrimination should be used to select them. Discrimination analysis can be done by the various techniques specially devised for such purposes. Numerical taxonomy is thus based on certain principles, also called as neo-Adamsonian principles. Now, the principles of numerical taxonomy. The first principle of numerical taxonomy is the classification system will be better if the greater content of information in the taxa and more the characters taken into consideration. The second one is every character should be given equal weightage in creating new taxa. The third one is for comparison purposes, the similarity between any two entities is considered. Correlation of character offers in the groups of organisms under study. Thus, distinct taxa can be recognized. The fifth one is phylogenetic conclusions can be drawn from the taxonomic structures of a group and from character correlations, assuming some evolutionary mechanisms and pathway. The next one is the science of taxonomy is viewed and practiced as an empirical science. The last one is phonetic similarity is the basis of the classification in the numerical taxonomy. Now, the steps involved in the numerical taxonomy. The number of major steps involved in the numerical taxonomy. The first step is the selection of operational taxonomic units, OTUs, and the second step is the selection of characters or attributes. The third step is the estimation of resemblance, and the fourth step is the cluster analysis. The cluster analysis is done by applying statistics to process data, percentage of similarities and percentage of dissimilarities, and after comparison of the characters in the last step, a phenogram is constructed indicating the relative resemblance and the differences between the various taxa. Now, the first step is the selection of operational taxonomic units. The first task in numerical taxonomy is to select the operational taxonomic units. The basic units in numerical taxonomy are generally called as the operational taxonomic units. They are generally abbreviated as OTUs. They can be an individual, species, genus, family, order, or even higher rank taxon. Comparison of OTUs of equal rank is always made in numerical taxonomy. In case of OTUs above the level of individual, adequate representation of various polymorphic forms is essential. For example, when a genera are compared, they should be represented by different species. Similarly, when families are compared, they should be presented by different genera and so on. Thus, it helps to study the intraspecific and interspecific variations. The second step in numerical taxonomy is selection of unit characters or attributes. Unit characters or attributes are the characters which are used in numerical taxonomy. According to Sokal and Smith, unit character is defined as a taxonomic characters of two or more states. Only the phenotypic characters are used as the unit characters. For example, the presence or absence of hair or bristle-like appendages in a grass spikelets. Large number of characters are to be selected from the operational units. It is recommended that at least more than 50 characters should be taken into consideration. The value of comparison becomes more stable with the increase in the number of characters taken into consideration. Moreover, characters should be used from all parts of the plants and all the stages in the life cycle of the plants. The taxonomic characters may be grouped into morphological, physiological, biochemical, ecological, and distributional characters. Now, the types of unit characters or attributes. Generally, in numerical taxonomy, two types of unit characters or attributes taken into consideration. The first one is binary characters and the second one is multi-state characters. Now, the binary characters. Those unit characters which possess two contrasting states are called as the binary characters. For example, the presence or absence of trichomes on the plants and the dehiscence or indehiscence nature of the fruits. The binary characters can be represented by the simplest form of coding where characters are divided into plus and minus or as 1 and 0. In such cases, the positive characters are recorded as plus or 1, whereas the negative characters are recorded as minus or 0. In case of the organ possessing in given character is missing an organism, the character is recorded NC, which means no comparison. Now, the second type of the attributes is multi-state characters. Multi-state characters are those characters which possess three or more contrasting states are called the multi-state characters. For example, the color of flowers may be different types. Say for example, the flower color may be red, it may be white, purple, yellow, etc. Such characters can be coded into the number of states like 1, 2, 3 and so on to the corresponding their ranges of the variations. The multi-state character or attributes may be further of two types, the qualitative multi-state characters and the quantitative multi-state characters. Now the first one is qualitative multi-state character. These character contains three or more contrasting form and each form is ranked on equal footing. For example, the flower color which can be in any number of state. It may be red, white, purple, yellow or of any color. The second one is the qualitative multi-state characters. These characters represents measure of the size on a continuous scale such as the weight or length. For example, the length of leaves, height of the plants, amount of the pubescence on a leaf of a plant, etc. Now, the third step involved in the numerical taxonomy is estimation of resemblances. Most phonetic methods involve taxon to taxon distance, similarity or dissimilarity measures. Distance and dissimilarity are sometimes treated as the same things, though a distinction can be made between them. As the name implies, distance and dissimilarity measure increase with the dissimilarity between taxa, while similarity measures decreases with the dissimilarity. Thus, the resemblance between two OTUs is estimated or measured either in terms of similarity, that is percentage of character in which they agree, or in terms of dissimilarity, that is the percentage of characters in which they do not agree. Now, the fourth step in the numerical taxonomy is cluster analysis. Cluster analysis or clustering is a type of multivariate statistical analysis. It is used to group organisms into separate clusters based on their statistical behavior. The main objective of clustering is to find similarities between organisms and then group into similar organisms together to assist in understanding relationships that might exist among them. Thus, different OTUs are grouped together on the basis of degree of similarity, and these groups of OTUs are called clusters. Cluster analysis is based on a mathematical formulation of a measure of similarity. Now, the methods of cluster analysis. The methods of cluster analysis were involved shifting of cells with similar value in the matrix so that the OTUs with closely related values were brought together in clusters. Today, with the advancement of computer technology, programs are available which can perform an efficient cluster analysis. Now, the types of cluster analysis. In numerical taxonomy, the data clustering algorithms can be the hierarchical clustering, and it may be further of two types. The first one is agglomerative clustering and the second one is divisive clustering, and the second type of the clustering is partitional clustering. Now the first one is hierarchical clustering. The hierarchical clustering techniques are an important category of clustering methods. A hierarchical clustering is often displayed graphically using a tree-like diagram called dendrogram. A dendrogram is a graphical representation of hierarchical clusters which are usually generated through a mathematical process such as cluster analysis. The purpose of a dendrogram is to display the relationships among distinct units by grouping them into smaller and smaller clusters as shown in this figure. To draw a dendrogram, first of all, we have to write down all the attributes or units or points in the bottom that will be in the dendrograms. Then draw lines to connect those units that are placed into groups only two. Not every unit will fall into such a group. Now here we have to make a grouping of two closely related taxa, closely related attributes or closely related units or points by drawing a line. So here I have make a group of point P2 and P3 by drawing a line and thus forming a group P2 and P3 group. Now again draw lines to connect groups of 3 or 4. This may involve connecting groups of 2, and here this group P2 and P3 is again connected by a line with a new attributes or another point that is P4, and thus forming a new group of 2 that is named as P2, P3, P4 group. And finally, we have to draw again a line to make the another group, and here this line forming a group of 2 P1 and P2, P3 and P4. Thus, we have here a final group of all points, all attributes and all traits into a single cluster that is referred as P1, P2, P3 and P4 group. Now, this is the hierarchical clustering of 4 points known as a tree-like diagram and known as the dendrograms. And here we have the another methods of the representation of hierarchical clustering is the nested cluster. So with the help of the nested clustering or nested clusters, we can illustrate the hierarchical clustering. Here, the closely related 2 points P2 and P3 are grouped by using a circle. Again, this group connected with the another closely related points with the help of another circles, and finally the next point may be encircled by another circles. Thus, such type of clustering is known as the nested clusters. Now, there are two basic approaches for generating a hierarchical clusterings. The first one is agglomerative clustering and the second one is divisive clustering. It displayed both the cluster subcluster relationships and the order in which the clusters are merged is known as agglomerative clustering, and in which the clusters are split into two and more individuals or taxa or units is known as divisive clustering. Now, here the first one is agglomerative clustering. The agglomerative hierarchical clustering techniques are the most common techniques which is being used in the numerical taxonomy. This method starts with T clusters equal to the numbers of OTUs. These are successively merged until a single cluster has finally been formed. Sequential agglomerative hierarchic non-overlapping clustering method SAHN is the most commonly used clustering method in the numerical taxonomy to achieving hierarchical classification. The agglomerative clustering starts with an individual and continuously combined into larger and larger groups. The different steps are involved in the preparation of the agglomerative clustering diagram. In first step, each data point is assigned as a single cluster. And here we have 5 data points. These are A, B, C, D and E. Now determine the distance measurement and calculate the distance matrix to merge the clusters. In next step, draw the lines to connect those units that are placed into groups of only 2. Not every unit will fall into each group. So here we have to make, in next step we have to make the grouping of 2 traits or 2 points. So here the grouping of, one grouping is there, the grouping of A and B, and the another grouping is there, the grouping of D and E. In next step, again draw lines to connect groups of these traits into 3 or into 4. In next step, they repeat the process of the clustering or grouping until every data point becomes one cluster. So now we can say the agglomerative hierarchical clustering starts from the single traits, single attributes or single OTU and finally grouped into a larger clusters. So now here the each data set or each attributes or OTU in agglomerative clustering are known as the leaves, while as the intermediate groups are known as the nodes, and the final cluster, the final larger cluster is regarded as the root. Now, the second type of hierarchical clustering is divisive clustering. The divisive methods, as opposed to the agglomerative methods, starts with all T OTUs as a single set, then subdividing into one or more subsets. This is continued until further subdivisions is not necessary. The commonly used divisive method is association analysis. This method is mostly used in ecological data employing two-state characters. This divisive clustering is also called as the top-down clustering. Thus, the divisive clustering method is opposite to the agglomerative clustering. It starts with all individuals or all OTUs and continuously splits into smaller and smaller groups. So here we can say the divisive clustering is just opposite to the agglomerative clustering. Here it starts with all characters, all traits and all attributes and all OTUs, and it splits into smaller, then again into the smaller groups. The different steps are involved in the preparation of a divisive clustering diagram. In the first step, all points, all characters in a data set belongs to one or single cluster. In next step, we will make a partition in the cluster into two. And in next step, we will make a partition in the cluster again into two and least similar clusters. In next step, we will have to proceed it recursively to form new clusters until the desired number of cluster is obtained. Thus, finally, we will get a divisive clustering diagram. Now, in numerical taxonomy, after the hierarchical clustering, the second type of clustering which is used is partitional clustering. A partitional clustering is simply a division of set of data objects into non-overlapping subsets or clusters in a manner that each data object is in exactly one subset. It is used or based on specifying an initial number of groups and continuously relocating characters among groups to convergence. Thus, we can say in a partitional clustering, simply a division of sets of data or objects into non-overlapping subsets and clusters. They may be single clusters, they may be two clusters, they may be more than two, 3, 4 and so on, many clusters. And here we have 4 clusters. The first diagram showing the single cluster where the single set of attributes and single set of data clustered in a single group, and while in this one there are 2 cluster formation takes place, and here we will find 3 clusters, 1, 2 and 3, and likewise in this diagram we will find the 4 type of clustering. Then this is the partitional clustering. The fifth or last step involved in the numerical taxonomy is the construction of a phenogram. The numerical phonetic methods organize the taxa or operational taxonomic units or OTUs into groups based on the overall similarity. OTUs are grouped in such a way that there is a higher degree of similarity within groups than between groups. It means two OTUs within a group share more characteristics than do two OTUs in different groups. The least inclusive or smallest groups represents the highest similarities. More inclusive groups share successively fewer characters states, and the most inclusive groups is the most heterogeneous, including all the OTUs in the study. A clustering method is usually used to group OTUs that are most similar. The agglomerative clustering algorithms are popular for this purpose. Now, the phenogram. Phenogram is a branching diagrammatic tree used in phonetic classification to illustrate the degree of similarity among taxa. This hierarchical pattern of grouping is expressed in a kind of branching or tree diagram known as phenogram. The relationship of OTUs and groups in a phenogram reflects only similarity. There is no evolutionary relationships are implied between two OTUs or two taxa. Thus, it is a dendrogram and it represents the graphical presentation of hierarchical clusters which are usually generated through a mathematical process such as cluster analysis. The purpose of a phenogram is to display the relationships among distinct phonetic characters by grouping them into smaller and smaller clusters as shown in this figure. To draw a phenogram, first of all, we have to write down all the phonetic units across a plane. And here there are 4 phonetic characters or units. These are P1, P2, P3 and P4. Then draw lines to connect those units that are placed into a groups. And here the grouping of 2 is formed by connecting P2 and P3 units, and thus these 2 units forming a group P2 and P3. Again, draw lines to connect groups of 3 and groups of 4. This may involve connecting groups of 2. So here this group P2, P3 again connected with the unit P4 by a grouping method by drawing a line and thus forming a group P2, P3 and P4. And finally this group may be connected with the another point P1 by drawing a line and thus forming a final or larger group P1, P2, P3 and P4. Thus in phenogram we can say we make a groups from the single unit of character of phonetic characters into larger and into largest groups. And here we can say the similarity increase towards this direction. Now, the applications and advantages of numerical taxonomy. The first one is numerical taxonomy can be successfully used in the study of various angiospermic genera such as Apocynon, Chenopodium, Crotalaria, Cucurbita, Oenothera, Salix, and the cultivars of wheat and maize crops. Next one is the similarities and differences in microorganisms such as bacteria can also be studied by the numerical taxonomy. The data of conventional taxonomy is improved by the numerical taxonomy as it utilizes better and more number of described characters. As numerical methods are more sensitive in delimiting taxa, the data obtained can be efficiently used in the construction of better keys and classification system. Thus, the methods of numerical taxonomy have been successfully employed to a wide range of biological organisms, and to a great extent they provide results that agree with established classification. Thus, they are highly relevant to the real problems of the biological classification. Besides the applications and advantages of numerical taxonomy, we have some disadvantages of the numerical taxonomy on the other hand. They are: the numerical methods are only useful in phonetic classifications and not useful in polyphyletic or phylogenetic or evolutionary classifications. The biological species concept may not accept the specific limits bound by these methods. Selection of character is the greatest advantage in this approach. If character chosen for comparison are inadequate, the statistical methods may give less satisfactory solutions. Different taxonomic or taxometric producers or groups may yield different results. The major difficulty is to choose an procedure for the purpose and the number of characters needed in order to obtain satisfactory results by these mechanical aids. This is all about the numerical taxonomy. I hope this lecture will be very fruitful to you. If you have any doubt and any queries regarding to this lecture, you may write down in the comment section. And thanks for watching this video.